Hello, all. Welcome to week 3 of animal biology. Uh, the cutoff for dropping the class without a W is today, as of recording, the 24th. If you want to get out of this class, get moving on it. Yeah, so I've been working with a lot of folks that are having issues with technology. I've been working with a lot of folks that are having issues with assignments. In general, reach out to me before the due date of an assignment to say that you're going to have trouble getting it in on time. Do not reach out to me after. The reason I make things due on Sunday is to give you a week to kind of troubleshoot and figure things out. Like you even have Saturday to go and work on it. Again, I am not available over weekends typically. This is due to health issues that I need to go and take very seriously. Uh, Past that though, yeah, I will continue working with you folks. Please double check the instructions if you are still having trouble with setup. And if you run into too much of a wall, contact your issues with Pearson, contact Pearson. If it's with just general tech setup, contact IT. They are more qualified to help you than I am. I'm a biologist. They gave me this class as a tool. I don't know how to fix the tech stuff most of the time. With that, I think it's about time for us to go and move on to our presentation. So I will start doing that. All right, so I'll just be in the corner here. I'll move us out of the way if we need to. All right, so we are going to be talking about chapter 21, DNA and biotechnology, this week in week 3. I don't know where I want to put me. I'm gonna put myself here for now. All right. So we're going to go and be looking at oh, give me a second. Have to wear Invisalign. It's a bit rough to go and be talking for too long with it. Alrighty, so we're going to be talking about the form of DNA, the replication of DNA, gene expression, mutations, epigenetics, which is the regulation of gene activity, genetic engineering, and genomics. So there are a couple different forms of DNA. The main form of DNA for eukaryotes, which is, well, okay, there's a couple different forms of ways that creatures convey genetic information between generations. DNA is the type that eukaryotes like us utilize. So DNA is a double-stranded molecule. Its arrangement should look like a ladder that somebody has then twisted by the rungs from either end. And it should be a string of repeating units. Each side is half of a rung, and they're made from nucleotides. Nucleotide is the monomer for the DNA's polymer, meaning that it is a single component of what makes up a long chain of a molecule. So which is the polymer. Think of the monomer as a Lego, and the polymer as the Lego Eiffel Tower. It's a component that makes a larger whole. So each individual nucleotide is made out of one deoxyribose sugar, one phosphate, and one nitrogenous, which just means it has nitrogen in it, containing base. DNA has 4 nitrogenous bases. These are adenine, guanine, thymine, and cytosine. RNA, which is a single-stranded molecule that is also used to convey genetic information, and it's the primary mechanism for genetic information for prokaryotes, replaces thiamine with uracil instead. Or it might be guanine. It shows up later in the presentation. We'll talk about it. All right, so rungs of ladders. So each rung of the ladder consists of a pair of nitrogenous bases. Each pair of bases are held together by hydrogen bonding, the same energy that makes water molecules like cohese to each other. And they have complementary base pairing rules. So adenine only pairs with thiamine, and cytosine will only pair with guanine. That means that if you know the nitrogenous bases of a single line of the ladder, like one half of it, you can go in with 100% accuracy, project what the other half is going to look like. Genetic information is encoded in the exact sequence of nitrogenous bases. So this is kind of what it looks like. Here's our phosphate group. Here's our deoxyribose sugar. and here's our nitrogenous base. So this little video will help demonstrate what I've been describing with some visual aids.
The building blocks of DNA.
Alrighty, so that video kind of elaborated a little bit on some of these concepts, let you visualize it, and also describes how new DNA is synthesized. It's important to note that every individual that comes from a species that sexually reproduces has one copy of DNA from each parent. So like I'll have one copy of like chromosome 22 that has the gene that controls like hair color on it. And I got one copy from my mom and one copy of my dad. So whenever we talk about DNA base pair, whenever we talk about your genes, we talk about them in terms of pairs in general. So here you can go and see the double helix model of DNA and its attached rungs inside of it. So DNA is composed of 4 nucleotides. We have base pairs, nucleotide, a nucleotide, and a phosphate. This is just helping you visualize the things we've described already. DNA needs to be replicated. Namely, we need exact copies before cellular division can take place. So both mitotic and meiotic cellular division require DNA replication. And this makes sense. If you have 2 copies of DNA, one from each of your parents, and you need those 2 copies of DNA in order to go and survive, if you were reproducing, you would need to double your DNA so that you and your offspring both can keep your DNA, right? Now, in DNA replication begins with an enzyme, which is just any sort of chemical that can go and enable a chemical reaction, or any sort of chemical that enables a different chemical reaction to occur without being consumed itself. So these things usually just lower the activation energy of a chemical reaction that they are not actually in. So what DNA helicase does is it unwinds the DNA strands by breaking the hydrogen bonds that hold together DNA base pairs. So right here, these dots are our hydrogen bonds. This causes the strand to split apart. So, all right. So as these 2 strands separate and unwind, the nitrogenous bases are temporarily exposed. So this is not something we want to happen for too long because these are very reactive molecules. And if they're exposed for too long, the DNA could start to get damaged. At this point, the free nucleotide bases, though, that form a new hydrogen bond with a complementary base pair, following the same code that we see here. T's will always bind to A's. If we split this in half, then then the free T is going to seek out any sort of A it can find. DNA polymerases then move through the strand that is composed of free-floating base pairs attaching to other free-floating base pairs, and reattach the sugars and the phosphate groups of the base pairs to where they're supposed to be attached, resulting in a new double-stranded DNA helix. So this is known as semi-conservative replication because one of the strands that is so the new double helix that you've made, the double strand of DNA, one is the original strand and the other is a new strand known as the daughter DNA that's made by those free-floating base pairs we were talking about. Complementary base pairing creates two new DNA molecules that are identical to the parent molecule. This leads to accuracy in passing down genetic information, and it means that a parent cell to a daughter cell, that genetic information has to be passed down from a parent to a daughter cell. We cannot spontaneously make cells. Is that process explained maybe a little better than how I was doing it? But you see, the molecule, the DNA double helix is unwound by the what's the name of it? Helicase. The lagging strand starts to get formed. The intact DNA strands act as a format for new base pairs to bind themselves to. And then the strand itself is repaired and patched by the action of other enzymes, resulting in two identical daughter double helixes. How does RNA direct cellular activities? DNA codes for RNA, RNA codes for proteins, and proteins play structural or functional roles in the cells. So we call this the hierarchy of protein synthesis, and it is one of the most important concepts in molecular biology. So before we go too much deeper, we need to explain the concept of a gene. A gene is a segment of DNA that codes for a particular protein. So I might have a gene that controls like the production of amylase in my saliva. It is coding for the protein amylase levels of production. And so we call that region of DNA the amylase gene region because it's coding for the amylase gene. When we say gene expression, we mean that a protein is produced. So when a gene's being expressed, a protein, that gene's DNA is being used in order to go and send out the orders to create a particular protein. RNA, ribonucleic acid, is different from DNA in a couple key ways. So RNA is a way of conveying genetic information and is the primary mechanism of conveying genetic information for a lot of microbial life, all the prokaryotes. However, RNA is single-stranded, which means it's significantly less sturdy than DNA. DNA is protected by that phosphate backbone, so RNA breaks down quicker, so you can't have as long-lived of animals. And it contains the base uracil, or can't have long-lived organisms, and it contains the base uracil, not thiamine. So this is a 1-for-1 replication. Thiamine is replaced with uracil in RNA, where thiamine would normally bind, a uracil binds instead. And again, the sugar is ribose, not deoxyribose. That's why it's RNA, not DNA. Here's a chart just showing their similarities and their differences. Both are nucleic acids, so they convey genetic information between generations. They are both composed of linked nucleotides being those base pairs. They have a sugar phosphate backbone, which we go back here. Here's our phosphate, here's our sugar. And they form the blue components of this model. So the blue spiral backbone here, we call this the phosphate backbone, but it's made of both sugar and phosphate. All right. So DNA, so they both have a sugar phosphate backbone, and they both have four types of bases, which means that these bases are read in three sequence codes. So that leads to a very large number of different combinations for the production of amino acids. Now, DNA is double-stranded while RNA is single-stranded. DNA has deoxyribose while RNA has standard ribose. And DNA, as a result, has the bases of adenine, guanine, cytosine, and thymine, while RNA has the bases of adenine, guanine, cytosine, and uracil as a direct substitution for thymine. DNA functions primarily in the nucleus, while RNA functions primarily in the cytoplasm. It is important to note that RNA is in every single living organism. DNA is only present in eukaryotes. So RNA can do all the jobs that DNA can do. DNA is just better at doing some jobs in particular, which is why it is the primary mechanism of storing genetic material for eukaryotes. So RNA synthesis, protein synthesis, occurs in two steps, and these are transcription and translation. So first, you start with transcription, you start by synthesizing the mRNA or the messenger RNA, using DNA as a template. So mRNA then carries DNA's instructions for synthesizing a protein out from the nuclear envelope to the cytoplasm. Interestingly enough, the nuclear envelope has nuclear pores in it, and those nuclear pores are only one strand of RNA wide. This is because the double helix DNA needs to be kept inside of the nucleus, so the pores are just too small for it to fit out, while the RNA can fit out. Steps in transcription. So DNA unzips along the hydrogen bonds using an enzyme known as RNA polymerase. Free RNA nucleotides then pair with complementary DNA bases. So, like, if there's a C that is being opened up by the unzipping of the DNA, a G is going to bind to it. If a U is opened up by the unzipping of the DNA, an A is going to bind to it. This forms an RNA transcript. This chain of RNA base pairs that are complementary to the DNA base pair chain. RNA transcript is then released from the DNA, and the DNA rewinds itself and closes up that helix again. So, the actual transcription sequence is started by a section known as a promoter. RNA polymerase binds to the promoter, and it unwinds the DNA helix and aligns and links together the RNA nucleotides. So it's building the backbone for the RNA nucleotides as well. What signals the strand of RNA to stop synthesizing, to stop copying the DNA, and to just terminate is a sequence of bases on the DNA known as a stop signal or a stop codon. A codon being a group of three amino acids that code a group of three nucleotides that code for a single amino acid. In this case, it's known as the stop codon yeah. So, all right. Oh God, what the? Da, 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 da. Sorry, I'm having trouble with the computer here. All right. So stop sequence for basis for the stop signal. All right. So a technology most days. So the segment of DNA to be found. So the segment of DNA to be transcribed unwinds. RNA nucleotides pair with the complementary DNA bases and are linked together to form an RNA transcript. From there, the RNA is released from the DNA. RNA polymerase is managing this activity. That's probably the best way to explain it. I think I was making it too complicated on the prior slide. So, modifications to mRNA before it leaves the nucleus. So, on the before the RNA sequence leaves the nucleus, a what is known as a 5 cap is added onto the end of the sequence. This is think of it kind of like an aglet on your shoelace. I learned that one from Phineas and Ferb. So, whenever this RNA it comes across a reactive molecule while it's traveling to the ribosome to start transcribing proteins, uh, this 5 cap will be damaged instead of the actual content of the message. The 5 cap is designed to be damaged, and in doing so, protects from degradation. There's also the other side is going to go and have what's known as a poly A tail, which also is degraded instead of the DNA sequence, but it specifically has a role in helping the RNA leave the nucleus. So, so oftentimes there are discontinuous sections of code in DNA. When this is copied over to mRNA, this unused section will be known as an intron. So an intron is not necessarily a useless section of the DNA. It might code for a different protein, or it might be a starting point for a different sequence of amino acids. But they will not be necessary for this process. So they're going to be snipped out by specialized enzymes to go and remove that intron that's not useful for this particular project. Exons, by contrast, are the expressed sequences. They are the whole point of copying over the DNA to the RNA. So this is going to be left to so these are going to be allowed to express themselves. They're going to be directly utilized for the protein synthesis. And this is what's going to actually leave the nucleus. So here you see DNA, RNA polymerase, and then the gene to be transcribed. Let's watch this video and I'll go and explain what's going to go on. Whoops, wrong button. All right. So that's just another a way of visualizing alongside the explanation. Here you can see the mechanism by which introns are cut out of the exchange. So here we have the exon, exon, and exon. We want to code for all these amino acids, but the introns are in the way. We don't want the introns to go and be part of the sequence. So a specialized enzyme removes these introns, leaving behind a new RNA molecule. Another enzyme then splices these exons together to make a continuous sequence. That is what is actually going to be leaving the DNA nucleus. So here's a review of the functions. So messenger RNA carries DNA information. Transfer RNA binds to a specific amino acid and transports it to be added as appropriate to a growing polypeptide chain. And ribosomal RNA, or the tiny speck-like organelle specialized for the synthesis of proteins. Oh, ribosomal RNA combines with proteins to form ribosomes. I'm sorry, I didn't finish reading the section. So ribosomal RNA is an essential component that is produced by a very specific section within the nucleus and leaves complete from this specific section of the nucleus, known as the nucleolus. So the nucleolus is the site of ribosomal RNA synthesis. Protein synthesis. So first, we start translation. mRNA gets turned into a protein. The nucleic acid is going to be, yeah. Then genetic code converts the DNA base sequence into an amino acid sequence. So RNA is the pro. I'm sorry, give me a sec. This computer is falling apart. So protein synthesis, mRNA is used to go and transcribe a code from the DNA that is then used to synthesize amino acids, which are then stuck together to go and form a protein. Proteins can be subdivided into 3-letter sequences, 3 base sequences. Each one will correspond to a single amino acid, which are the building blocks of proteins. An example of this would be UUC on mRNA, specifies that once it reaches the ribosome, it's going to synthesize the amino acid phenylalanine. There are 3 different stop codons that signal the end of a protein. These are UAA, UAG, and UGA. They do not code for an amino acid. So similar processes were followed for every single amino acid that we know of. We were able to go and figure out the code for each one. So this chart is designed to go and help you find those codes. So to read this, you start over here on the first base column. So you start over here on the first base column, which then helps you pick the first of your 3-letter sequence for your codon. You then move. So let's say we want to go and check out C. The first C in our sequence is C. Then we're going to go and move over to this column or this row. So our second letter is going to also be C. So then we move over to the third row. So within the C section, for the C row, the third letter is G. So that means that we're coding for CCG or proline, the proline amino acid. So then we have transfer RNA. So tRNA converts genetic messages into particular sequences of amino acids. So it can recognize the codon on mRNA and what amino acid is specified by the codon. Enzymes ensure that tRNA binds with the correct amino acid, and tRNA then ferries the amino acid to the correct location along the mRNA strand. So it would be incorrect to say these amino acid base pairs are just floating around in the void. They are guided. Now tRNA knows the correct mRNA codon to bind to because it possesses a 3-base sequence that is complementary to the mRNA codon known as the anticodon. Here you can go and see how that kind of works. So we have our amino acid phenylalanine, the sequence for it, AAG, and the mRNA that it's going to be anchoring itself on down here. So this is a opposite complementary sequence, and this is how the actual amino acids are getting involved into the situation. So ribosomes have 2 unique subunits, each composed of ribosomal DNA. One is composed of ribosomal DNA and one is composed of a specially folded protein. The purpose of a ribosome is to bring tRNA next to mRNA so that they can then bind to each other. This leads to a permanent alignment of the anticodon and the codon. Ribosomes are the only organelle present in prokaryotic organisms, and they are the smallest organelle composed only of 1 folded proton and 1 chunk of RNA. So here, in a way, an RNA acts kind of like a giant enzyme. It's enabling a reaction to occur by bringing the right components together in the right way, but it is not consumed itself in the reaction. All right. So there are 3 stages of translation. The first is initiation. So major players in protein synthesis come together. These are mRNA, your tRNA, and your ribosomal subunits. So here you can see, wow, this is the most annoying feature of Zoom, isn't it? There we are. So here you can see the large subunit with the 2 tRNA binding sites on it, moving into position over the top of the small subunit, which is guiding the mRNA over its length. So this occurs at AUG because that is our start codon. The large subunit then slots into the small subunit and begins anchoring amino acids onto this chain. After that, we engage in what is known as elongation. So enzymes in the ribosome link the amino acids to each other, slowly growing chains of the amino acids, slowly synthesizing our proteins. This occurs all along the length of the mRNA, and there are many clusters of ribosomes that interpret it at once. So this speeds up the process. This complex of ribosomes is known as a polysome because there's many of them. Poly just means many in Latin. Here you can see the process followed through. So tRNA with appropriate anticodons pairs with the next codon in the mRNA, and then it anchors and it slowly feeds out our polypeptides, these blue beans, as they get longer and longer. The RNA with appropriate anticodons pairs with the next codon on the mRNA. Enzymes link the amino acids to each other. The tRNA in the first binding site leaves the ribosome, and then the ribosome moves on to the next mRNA, exposing the next codon. And then the process just keeps cycling over and over again until you hit a stop codon, at which point it is expelled and released. Termination is what we call this process. That is when the stop codon moves into the ribosome. So the stop codon moves into the ribosome. This triggers release factors, which tells the ribosome to reopen, releasing the mRNA. Here's this process visualized. There you go. So that's the process I described, just, described clear. All right. So mutations are random changes in the DNA. So there are a lot of different types of mutations. One of the first is chromosomal. So this is where a full section of the DNA is duplicated or deleted. So mutations can occur for many reasons. They can occur because of radiation bombardment, chemical bombardment. That's what a carcinogenic is. Errors in the division of a cell, sometimes induced by too rapid of division. Most mutations just result in the destruction of the cell that mutated. Some of them give you cancer. In general, though, this is the main mechanism of random change that's occurring from in asexual reproduction. So if you have sexual reproduction, you can have allelic reshuffling because genes are randomly assorted from each parent. But when you are doing non-sexual reproduction, in theory, you're a clone of your parent. So the only changes that can occur have to occur during mutation. So uh, so some of the ways that your DNA can be changed by mutation is the order of the nucleotides can change. There can be insertions or deletions of nucleotides. And as a result, the final protein can be completely changed or completely destroyed. So for reference, here we have the original DNA and then an incorrect pairing on the DNA. So this gives us two possible outcomes, because they no longer match. So one DNA is identical to the parent. So this one is going to code the same as the parent would. It's going to code for valine. The other strand of DNA no longer matches the parent, and so it's coding for in a completely different amino acid, alanine. So epigenetics is the study of gene activation. So normally only certain genes are active in a certain type of cell. Like we have the full DNA for our body and every one of our cells, but my skin cells do not produce insulin. My liver cells do not produce hydrochloric acid like my stomach cells do. They have the full genomes, but only some of them are being expressed. This allows for cellular specialization, and it also allows for your body to make changes within your lifetime. So if I get assigned a job working in the valley and I'm working in the sun all day, I'm probably going to get tanner and I'm probably going to get a bit thinner. So my DNA is not changing, allowing me to have darker skin and be thinner. The genes associated with that behavior are being expressed because the environment has triggers in it that is triggering that expression. The DNA has always been there. It's just being triggered finally. So active genes produce specific proteins that determine the structure and function of a particular cell. So that's just the active genes, the ones that aren't being activated by outside triggers. Gene activity is regulated simultaneously at several levels. So at the chromosomal level, your DNA is constantly coiling and uncoiling, except so which is the one of the main mechanisms, which if DNA is being coiled, it cannot be replicated because it cannot be accessed to go and be transcribed. So that's used as one of the regulatory mechanisms to determine what amino acid is being expressed when. So then we have the transcription of genes. So certain genes have an increased rate of transcriptions, and certain hormones are able to turn on certain genes. So a hormone is any chemical that signals from one cell to another through a chemical action. Steroids are signaling hormones that are based off of lipids or fats. The sex hormones are a subset of steroid hormones. These include like testosterone and estrogen, that sort of thing. Now, the School of Biotechnology is the manipulation of living cells to perform specific tasks. So, biotechnology tends to focus on the microsystems and not the macrosystems. Like, if you get much bigger, now you've transitioned into animal science. If you get bigger than that, now you've transitioned into ecology. So, biotech is the manipulation of cells to produce to perform tasks. So, like, they'll make a bacteria cell that produces insulin as a waste product, which then can be marketed. Genetic engineering is a subset of biotech that manipulates DNA specifically for human purposes. So sometimes you might just go and develop this new insulin bacteria by selectively breeding, like the traditional techniques by which we've bred everything that we've domesticated over the last 10 to 20,000 years. So genetic engineering is much, much newer. It is the mechanism by which we directly manipulate the DNA to change expression. One of the more common examples of this is the vast majority of crops on the US market, including the majority of organic crops, are actually less than 100 years old. So those crops were synthesized by putting those plants onto the deck of an aircraft carrier within range of nuclear testing. Those plants were then bred and grown to term to see if any favorable mutations had occurred due to the radiation exposure. So if you have somebody who's anti-GMO, like anti-genetic manipulation, they're going to have a hard time because every single crop in the US is genetically modified due to this period of history where we used radiation as a tool to change their genomes. But I'll tell you, as a biologist, genetic modification doesn't really negatively impact anyone. Like when we modify a genome, we are just doing the process of selective breeding like we've always done, just a lot faster. And if the animal survives to adulthood, or the plant survives to adulthood, it's going to go and be we'll have a very reliable record of how those traits are going to be manifesting. And as a result, don't fear don't be anti-GMO. Genetic modification is the only reason we can feed everyone. Be nice. The production of drugs and synthetic hormones are a direct result of genetic engineering. We can actually produce like testosterone and estrogen using bacteria now. This has improved our treatment of human diseases, especially a lot associated with geriatrics. Sex hormone levels tend to go and start to fall off when you get older, and it can lead to a lot of skeletal issues. And of course, it has directly influenced our food supply. Every single crop we eat now has been genetically modified either through selective breeding originally or radiation exposure or guided genetic manipulation. Like you're not going to get around. So recombinant DNA, also known as chimeric DNA, is DNA from two or more sources. So we do this pretty commonly to try to go and improve the immune qualities of plants. So like apples will occasionally have plum genes inserted into them because it makes the apple immune to a certain disease that affects apples. But because we're only transferring over the immunological genes, it does not influence taste, texture, or anything else like that. Yeah, so this is one of the mechanisms by which we replicate DNA in mass. This is, I believe, the main industrial mechanism by which DNA is replicated in mass. So, more you know. So, restriction enzyme. So, in order to go and create recombinant DNA, restriction enzymes cut out your gene of interest. Then you have to splice it into your vector DNA. Ooh, sorry. I need to eat less acids. My gut is not happy. So, so DNA, so it's important to note that circular pieces of DNA don't really tend to exist very often in nature. So RNA tends to be circular, and it is the genetic coding that is inside of prokaryotes. But we can go and splice in DNA, and it will be duplicated. Which isn't a, which, or we can splice in an RNA sequence, and then we can use that as a template strand to create DNA. So steps, yeah. Yeah, it doesn't need to be incubated inside of a living bacteria. Honestly, I feel like these slides are just doing a worse job of explaining what was from the video, so I'm gonna move forward a little. Yeah. Oh, you do need to go and do some regulation when you're running a project like this. If you have a you you have to go and double check to make sure that bacteria that were not containing the DNA did not successfully reproduce. So that could have been a result of a mutation or contamination, but you do need to go and sequence the DNA of your product. So PCR, the polymerase chain reaction, is the name of the industrial process that we use that takes advantage of this phenomenon of bacterial cloning. Alrighty. So, oh, I'm sorry. I hit the space bar. The video, we can't do anything. Okay, yeah. So that this was pulled from a lab activity. That's why it got confusing. But the point is, we use this mechanism in order to mass produce DNA for different studies. PCR is one of the first things you learn in a biotech in any sort of biotech class, and it's a very important process. Here are the steps for PC. Here are the steps for DNA replication through the use of cloning bacteria, and this is hybridization of the DNA. And here's what's occurring on the actual DNA level, as opposed to on the cellular level. And this is the and again, this is the termination of that cycle. So so there's a lot of applications for genetic engineering. One of the biggest ones that you always hear about are the oil eating microbes or plastic eating microbes. Uh, we've had success developing bacteria that can do both of those things. The problem is they cannot survive outside of laboratory conditions. Otherwise, we probably would be dropping it out of helicopters and airplanes and things like that in order to melt all the plastic that's everywhere. We've also used it to genetically modify our livestock. So the production of our livestock has quadrupled, as has the production of most of our plants over the last 100 years. Because of genetic engineering, it's been super helpful for us, and it's the only reason we can feed the world. Uh, so certain genes or certain valuable substances can be synthesized inside of organics. These would be things like uh, insulin, different types of enzymes, different types of proteins. Uh, so we'll occasionally go and modify the genes of an animal or a microbe in order to produce this stuff. There are bacteria that produce insulin. Previously, we'd modify horses to produce human insulin, stuff like that. One of the older projects that I heard about, like this was when I was like, I don't know, I'm 28 now. I remember hearing about this when I was like 6. They have modified goats in order to go and produce spider silk instead of milk. So this project was pretty much useless. We don't really need industrial amounts of spider silk, but it was a proof of concept because now they're trying to figure out how to do it with silkworms so that they could use goats to directly compete with the Chinese silkworm market. I personally don't see it going anywhere. Quite frankly, silk isn't high enough value to justify this. But if we had done this during in the 1500s, it would be a really big deal. Oh, then we also constantly modify crops in order to be resistant to pests. We do the same thing with domestic animals. The current domestic Holstein has like is exponentially more productive, exponentially more energy efficient, and exponentially more immunologically sound than its ancestors from even 60 years ago. This is how the spider silk thing worked. So we injected the gene of interest into a goat. In this case, this could be a spider gene or this could be an insulin gene, it could be anything. We implant the cell into the host mother that then has offspring that have this modified gene. For this one, let's say we wanted them to produce insulin in their milk. So we then collect the milk, refine it to isolate out the insulin, and we've synthesized insulin from a goat. You now have a goat that if you send the milk to a hospital, they can spin down essential medication for people. Gene therapy is a newer and at this point not super effective medication. So the goal is to go and take people who have genetic diseases, genetic dysfunction, and replace their fucked up genes with a healthy gene. Unfortunately, it's kind of hard to go and do this because we need to diagnose it before the individual is an adult for the majority of diseases, ideally before they're even born. And we have to be careful because the mechanism for delivering this DNA into their cells is to take a modified virus and have it infect their cells. So that needs to be done very carefully to make sure that it's safe. However, if we succeed at this, we could go and cure, or at the very least effectively treat, many genetic illnesses and conditions you might not think of as genetic that do have genetic linkages, like autism. Autism's more likely to occur along genetic lines. If it can be corrected, it might be a condition that can be cured. The other examples of this usually are like chemical synthesis issues that people have. So like insulin and diabetics are one of the more common ones. Multiple sclerosis. There's a disease that causes you to produce mucus inside of your lungs. We've effectively cured that with gene therapy if you can afford it. So yeah, here's a couple different examples. Healthy form of the gene into a virus. Modify the tissue by infecting the tissue with the virus and then reinsert it into the person's body. So this would be used to treat like leukemia. They remove all the marrow that is possibly diseased and replaced it with cultured bone marrow that's healthy. We can do so the study of genes is known as genomics. A genome is the entire set of DNA carried by a single individual of the species. So like the Human Genome Project was done in the late 90s, or I believe, and we sequenced all the DNA in a human, and we found out that there are 25,000 genes. To this day, we have figured out how maybe 5% of them work. Sometimes the biggest thing that we're realizing is that there might be emergent properties above the protein level for how DNA behaves. There's not a lot of we are not a very genetically diverse species. This is due to two near extinction events that we underwent. One was 100,000 years ago and one was 5 million years ago. Both times the human population or the progenitor species population dropped below like 8,000. That's really bad for genetic diversity. So humans are more closely related than almost any other species of animal. We have less genetic diversity, which leads to a couple unique conditions and health issues from the resulting inbreeding. So, epigenome. So these are chemicals that bind to specific genes to turn them on or off and are variable through life. It's impacted by diet, stress, sun exposure, exercise. This is how your body says you need to get tanner, you need to gain weight, you need to lose weight, you need to gain muscle. It's pretty much just the chemicals that interact with your genome to make it react to the environment around you. Then we have microarrays, which are tools used to analyze genetic activity under different conditions. So what they'll do is they'll take a glass chip and put thousands of identical DNA sequences onto the chip. And then they will analyze it under different conditions, occasionally using computer models, which helps us identify the function of different genes. This is where most of that 5% I mentioned earlier was discovered through the use of tools like this. One of the main things that we can actually use the genome for, since we haven't decoded exactly what everything means yet, even if we have decoded the genome, is it gives us an incredibly accurate record of how closely things are related. So taxonomy is the field of organizing life. And in recent years, we have pretty much thrown away all the old taxonomies in favor of a DNA model, and it has really cleared up a lot of stuff. one thing we've really realized is a lot of DNA is common to all life. So, for instance, we share 90% of our genes with mice. They're one of our closest relatives. The rodents are very closely related to primates. And we share 50% of our genes with fruit flies, showing that that is a surprising amount in common. Yes. And this is how, so these are different chunks of genes, and we are seeing how they are expressed in comparison to each other in a patient that has prostate cancer and a patient that does not. So you can see that prostate cancer tends to manifest in an overexpression of these tissues or an overexpression of these genes. If you see orange, is bad. Okay, so within this framework, I understand this is one of our most difficult subjects we will be covering this semester. I understand that, and I understand this stuff's hard. My first piece of advice would be to check out Crash Course Biology. They do a great video on transcription and translation. And my second piece of advice would be to go and review the study guide and all the reading. So within the framework of this lesson, though, our learning goals are as follows. You should be able to draw the structure of a DNA molecule and explain why complementary base pairs ensure reliable replication of the genetic code. You should be able to describe the purpose and mechanism of DNA expression, and you should be able to compare the structure and function of DNA and RNA, then describe the details of protein synthesis, including every stage of protein transcription. So if you can figure out how proteins are made and you can figure out how DNA is made, you're 99% of the way there. Again, Crash Course is an incredible resource. It's made by the guy whose brother wrote Fault in Our Stars. Very clever man. All right, let's stop that there. And I think that will cover it. You guys have a great day. Bye-bye.